In this video, I'm going to continue talking about GWAS, or genome-wide association studies, and I'm going to do so using an example from the literature. I found this paper because it seemed like a lot of y'all did some nice autumnal activities over fall break, like going to apple orchards and pumpkin picking. So I thought, let's find one has to do with apples. So this study is a genome-wide association study for the detection of genes related to apple Marsanina blotch disease resistance in apples. And I'm not going to try to pronounce that disease name again. I'm just going to call it AMB as the authors do in the paper. So the goal of this paper, as is stated in the title, is to use genome-wide association to find genes related to resistance of AMB in these apples. Let's briefly go over the methods of this paper, or what did the authors do? The first thing they had to do was look at their apples, that is, phenotype them. What is the phenotype you're observing? So they looked at trees and they looked at the leaves and saw over the period that they were monitoring them, were they apparently resistant to A and B? That is, they did not develop any of the blotches associated with the disease. Were they susceptible or were they very susceptible? They actually used a 6-point scale to tell this, and they did it based on the disease index, or kind of the severity of the disease on that tree. And next, they have to genotype the apple trees, that is, see what DNA is there. So for 192 different trees, they extracted DNA from the leaves. Next, they performed a restriction enzyme digest of that DNA, which followed by ligating some adapters and filtering and doing a size selection resulted in the reduced representation of the genome that they then submitted for sequencing. And in this case, they used Illumina to obtain short reads that they then mapped to a reference genome for the Golden Delicious apple. After they had their mapped reads, you can refer back to the RADseq video with the tuna if you want to know what I mean by mapping. And then, of course, the next thing, just like with the tuna video, they called variants. So in their reads, how were the sequences that they saw in their reads different from the reference genome and from other members of the 192 trees that are in this population that they have sequenced? And here I'll just quickly summarize the results of their sequencing effort. So total across all 192 trees, they got about 2 billion base pairs worth of data. And this represents about a 1.75% coverage of the genome. Remember, this is reduced representation genome. You're not looking to get 100% genome coverage. You just want a small fraction. And of that small fraction that they sequenced, the average depth was about 20. And the goal here is to look for genomic variants, or SNPs. So how many did they end up finding? Well, they ended up finding a whole lot, but then you filter them for just the ones that you see often enough in every individual. You may have to impute some missing genotypes, and also that are variable enough in the population. That is, you're really not interested in a SNP that doesn't show up, that only shows up in one or two individuals. So after their filtering steps, they found about 43,000 SNPs. 43,000 is still a lot. That's a lot of SNPs. And you're probably thinking to yourself, how am I going to look at 43,000 SNPs? How am I going to analyze that as an association? So just brace yourself for this next image, which is going to look like a lot, but we're going to walk through it. This figure is called a Manhattan plot, and this is often how scientists display GWAS results. It looks like a lot because it is a lot. You're looking at 43,000 SNPs right now. Let's go through this slowly. The first thing that you should note is that every single dot that you're looking at is a SNP. Now let's look along the x-axis where you see chromosomes. You're seeing the 17 chromosomes of the apple genome. So I'm just going to draw one over here. Here's our chromosome with the sloppy centromere in the center. So what you're looking at is that chromosome turned on its side and placed and squished up really tiny, so that the actual coordinates of the SNP that you see along the x-axis line up with the actual position of that genetic locus along the linear length of the chromosome. And this process is repeated for all of the chromosomes in the genome. The order could be anything here. Remember, within a chromosome, the order matters, right? Because SNPs that are closer together on the chromosome are actually closer together to one another. But a SNP on chromosome 1 and a SNP on chromosome 2, the distance between those is meaningless because they're unlinked. They're on different chromosomes. A SNP on chromosome 1 is no closer, you can't see the air quotes that I'm doing right now, but closer to a SNP on chromosome 2 than it is to a SNP on chromosome 3. So the order that we've placed these in is just numerical order. But you can imagine that you could switch it up if you wanted to. And then another thing that's been done just for ease of you looking at these data, and this is typically done, is that we alternate the color of the SNPs by chromosome so that you can clearly see where one chromosome ends and where another begins. So, to summarize, on the x-axis, you're seeing the physical location of the SNPs along their chromosomes. On the y-axis, you're seeing a value here called negative log 10p. That's the p-value that we talked about in the previous video. That's the p-value of the association test. But do you remember how the smaller p-values suggested a stronger association? Well, this plot would look really funny if we cared about the small values, right? It'd be hard to see. So we've done a mathematical trick here by taking the negative log base 10 of the p-value. And what that does, you can do the math if you don't trust me, is it turns a very, very small number into a big number. And again, this is just so it's easier for us to look at. And you can see this red line that's been drawn at about negative log 10 of 5. 5 would be between 4 and 6. And what that corresponds to is a p-value of 0.00001. The authors have provided that red line to show us, the readers, the significance threshold. So that is the cutoff above which they're going to be really interested in that SNP as associated with the disease phenotype. So what we have to do next to analyze these data is to see which SNPs are above that line. That is to say, which SNPs are significantly associated with the trait. And we see one here on chromosome 3, we see two on chromosome 9, one on chromosome 10, and one on chromosome 17. The next question is, what are those SNPs? Are they close by any interesting genes? Here are those SNPs in a table form in this paper. So again, we had the two on chromosome 9, one on chromosome 17, one on chromosome 10, and one on chromosome 3. You can see their physical position on this genome assembly, the p-value of their association with this blotch disease, the alleles that you see at each locus in the population, and then via BLAST or some other way that they used to annotate these genes, they found a candidate gene ID with its predicted function. A couple of important things that I want to point out here is that these are significantly associated, again, just like in the previous video, we're not saying that they cause the disease or they cause the resistance, but they are associated with this trait. And this is the predicted function of adjacent genes. If you remember how RAD works, you're only getting a very, very small percentage of the genome. I'm going to illustrate that here. So we have our complete genome in yellow, and I'm drawing a red block to indicate the actual gene. This is something that scientists don't know, but let's say that I do know it because I'm a magician who knows what gene is actually involved in this disease susceptibility or resistance. But our RAD tags, the places that we sequenced, remember we only sequenced about 1.75% of the genome. So the authors did make a note that their sequences were evenly distributed throughout the genome, except near the centromeres where things get tricky. But notice here that in the diagram that I've drawn, you don't actually get a RAD tag in the actual place where this gene is. But you do get one that's close by. You get two that are close by, right? So these are adjacent to the candidate gene, and they can still show a signal of association. And if you remember why that is, Remember the basic principles of chromosomal recombination and inheritance. Another way of saying it is that this rad tag and this actual gene, any variants that might be contained within this gene, can belong to the same haplotype in your population. That is, these variants, the one that you actually sequence and the one that you didn't sequence but is actually biologically meaningful, are linked to one another. Therefore, when you see variants at one, you're going to be detecting a phenotype associated with a variant at another adjacent location on the chromosome. And lastly, I want to say that even though the authors here only presented p-value, sometimes in a Manhattan plot on the y-axis instead of p-value, you'll see something called LOD or LOD, and that is the log odds ratio that we talked about earlier with the association test. And these authors didn't report it, but they did say that the minimum value was 3 for that odds ratio, that odds association. This value is actually pretty important to report. I do wish they had included it in this table here, because sometimes if you have a large enough sample size, you'll see a really significant association. But then when you look at the strength of that association, that is that odds ratio score, it's teeny, teeny, tiny. So it's only actually explaining a tiny, tiny amount of the variance in whatever trait you're looking at. So here it could say only explain maybe 1% of the variance observed in resistance to this disease. So when you are interpreting these results, make sure that you are actually thinking about that odds ratio as the effect size of what you're seeing. And the p-value is just the statistical support for that association. Now, I want to go back to the Manhattan plot for 1 second, just to make something absolutely clear. So let's zoom in just on this region of chromosome 3. And now remember, what we're looking at along the x-axis is that entire chromosomal sequence and all of the variants that we detected in our RADTAG sequencing. Now, there is somewhere along this chromosome an actual region, an actual gene, which I'm going to indicate with a little star there, that is actually involved in the resistance phenotype, let's say. Again, this is not something that I would actually know as a scientist, but if it were there, I could detect a signature. And I sequence rad tags all around it. So you'll notice something in the plot up here, which is that I should probably change the color of my pointer when we get a blue arrow. So we have the one locus that I'm pointing to right now, which is actually above our significance threshold. We are not saying that that locus, that SNP, is actually in our causative gene. We're just saying that it's probably nearby, that it is associated with the trait, and it's because it's linked to a gene that is associated with the trait. You might notice something that this one actually isn't as obvious as some other Manhattan plots. You'll see some Manhattan plots where it's like a big stripe of dots just shoots up all at once. That's because this SNP and this SNP are very close to one another along the chromosome. So you can kind of think as the one that's closest to the actual gene as pulling along all these others for the ride because they are physically linked to one another. They're close, like physically close to one another on the chromosome. That is, they don't get broken up by recombination that often because they're so close to one another. The probability of a recombination event happening between them is very low. So this is just me for the 1100th time saying that the loci that we're finding, the SNPs that we're finding associated with the trait may not be in the actual causative gene, but just physically nearby. And we can still detect that signature based on the general properties of recombination.